Well, here we are, and we're going to move on this time into looking at antibacterials and antivirals. This will cover the second half of D2, where the first half looked at penicillin. So remembering that D2 is looking at pharmaceutical products or drugs that are derived from natural products. So we'll look at penicillins in this case in terms of antibacterials, and we'll also look at antiviral agents concentrating on influenza and HIV over the next couple of videos. So essentially, these are very important aspects of drug design, with antibiotic resistance and viral conditions becoming some of the biggest health threats to our society. So let's have a look. So essentially looking at our understanding and applications and skills, first of all for D2 where we look at penicillin, we need to understand that penicillins are antibiotics and that they are produced by fungi. That the betalactam ring is part of the core structure, and we'll look at that as a particular structure that you should be looking at highlighting in your data booklet. We'll look at how some antibiotics work, how we modify those compounds in order to increase their resistance to different enzymes or to increase their potency. You need to be able to explain the difference in structure and the reproduction of viruses and bacteria. So contrasting the differences between viral diseases and bacteria. You need to be able to recall, which just means you have to be able to give a definition or answer simple questions in relation to this, that essentially antiviral medications are a newer class of drugs that are used to destroy viruses after infection compared to vaccines, which are used to prevent them. So antiviral medications after infection, vaccines are to prevent infection. You also need to be able to recall that antiviral drugs may work by preventing entry into the host cell, interfering with production of new viruses, or by preventing viruses from leaving the host cell. So we'll look at the modes of action or the mechanisms under which antivirals work. We will look at influenza as a case study for antivirals, and we will also look at HIV and the AIDS pandemic also for a target for the development of antiviral medications and how this affects the disease as a whole and how it is affecting populations around the world. So let's get into this and look at antibacterials and antibiotics. So essentially, antibacterials are drugs that kill or inhibit the growth of bacteria. Okay, they are antibacterials. So they are all about reducing how bacteria proliferate and controlling infection due to those. So an antibiotic, okay, and antibacterials are often used interchangeably. They are different things. Okay, so compounds that kill bacteria or microorganisms that are derived from other microorganisms are antibiotics. So penicillin would be an antibiotic. Okay, it is derived from a fungus, and its purpose is to kill other bacteria or microorganisms. So the penicillin group of compounds are produced by fungi and therefore kill harmful microorganisms, making them antibiotics, whereas antibacterials are any compounds, synthetic compounds that are used to destroy bacteria. So bleach or disinfectants, things like that, would include those, but compounds such as prontosil, which is a sulfonamide, which is a sulfur-based compound, are known as antibacterials. Okay, so diseases that arise due to the presence of microorganisms, particularly bacteria, okay, include things like tuberculosis, syphilis, cholera, salmonella, which gives rise from food poisoning, bronchitis, where you get the chest infection, anthrax, meningitis, gonorrhea, and chlamydia. So a number of sexually transmitted diseases, as well as infections of the lungs, are quite often bacterial based, and also of the blood. So let's look at penicillin. So a lot of you will have seen or talked about penicillin and how it relates to being a serendipitous discovery in terms of TOC and the nature of science and what we've just found out. So there were a number of scientists involved in bringing penicillin to where it is now. One of them being Howard Florey, who you might have remember that there is the Florey Institute here in Melbourne. He was an Australian scientist. So let's have a look. So Alexander Fleming discovered penicillin by chance. So he is the scientist who discovered it by serendipitous discovery. He was investigating bacterium, but didn't want to pursue finding out what the active ingredient was. So he left a bunch of petri dishes, came back, discovered that was mold growing on them, and where the mold was, the bacteria had ceased to grow. There was a zone of inhibition around the fungus. He didn't pursue that though. But Howard Florey and Ernest Chain were able to isolate the compound, which was penicillin G, from the mold and demonstrate its effectiveness as an antibacterial agent. So that discovery from Florey, so Fleming, original serendipitous observation, continued the research by Howard Florey and Ernest Chain to discover penicillin. Okay, but it was limited by how we could extract it. So Chain in particular was able to grow large amounts in corn steep liquor, so rather than through the fungus itself, which allowed us to have large amounts of penicillin. They were awarded the Nobel Prize in Physiology and Medicine in 1945, and the discovery of penicillin and its widespread use really made the difference, particularly in treating wounds in the Second World War. So this is essentially what it looked like. So you can see here, this was the bacteria spread onto an agar plate where the bacteria colonies are growing, and the plate got infected with a mold, a penicillin mold. And around that mold, we can see that the bacteria did not grow. So that's, and it was a compound excreted by these molds that was found to be responsible for that behavior. So the penicillin structure, all penicillin compounds share a common structural feature. Okay, so the wide scale use and production of penicillin was around from 1941, but we didn't have the structure until after 1945. So the structure of penicillin G, okay, was resolved by Dorothy Hodgkin using X-ray crystallography. And X-ray crystallography is a technique that we will look at that's used for elucidating structures a bit later on when we do topic 11. But essentially, it is like a dipeptide, so two amino acids coming together, but they come together to form this special beta-lactam ring, which is this square structure with the double bond O coming off here. So that ring is particularly relevant to penicillins. It's critical to their behavior, and up until this structure of penicillin was isolated, it didn't actually exist as a functional group, but we see it now in some other compounds as well. So penicillin activity is critical on this beta-lactam. So the compounds that penicillins are part of are known as beta-lactams because of this structural feature. So this strained square ring that we see in here with the nitrogen in it. So this is our natural penicillin, and then these penicillins are ones that have been created semi-synthetically, so modified from the natural product over time. So they're different forms. You will notice though that the general structure of them containing the beta-lactam ring is consistent across all penicillins. Okay, so they are susceptible to enzymes produced by bacteria known as beta-lactamase, and this enzyme breaks down this ring, which stops the penicillins from working. Okay, so the beta-lactamase attacks the penicillins here and can actually break them down. Okay, so but beta-lactams themselves affect the bacteria by stopping the cell wall from properly forming. Okay, so when this happens, so the beta-lactam binds onto the bacterium. As the bacteria, when it undergoes binary fission, and the new cell wall is formed, the penicillins stop the formation of the new cell wall. So you end up with holes in your new cell wall, and then due to osmotic pressure, water rushes in and bursts the new bacterial cells, so they undergo lysis. So let's have a look. We have a normal bacterial cell wall here, okay, which has cross-links. Then they grow up until they're ready to undergo fission. The penicillin now interferes with the linking enzyme, so it attaches to the cell wall, preventing new cross-links from forming. So these penicillin units here prevent new cross-links from forming, which weakens the structure of the cell wall. So the cell will burst from osmotic pressure because the integrity of the cell wall is not maintained, okay, when it goes to grow. This is all based on the reactivity of this beta-lactam ring, okay? So because it's highly strained, it's very primed to react. It's very reactive. So these ring structures mean that they are ready to pop open and behave in a certain way. So the beta ring structure of penicillin has a lot of ring strain. We've talked about these angles being strained around to 90 degrees, okay, and that allows it to react quite quickly with the bacterial cell wall. So in the active form, this ring is primed, it's strained. Beta-lactamase actually works by breaking this ring and making the penicillin inactive. So the bond angles have been forced to a non-standard bond angle. Okay, so usually we would see 109 and 120. What we see with this strained ring is bond angles around 90 degrees, which is what is responsible for the ring strain. The amine group will react irreversibly with the transpeptidase enzymes, okay, present in the bacterial cell wall, which are critical for wall stabilization. So it causes ruptures in our cell wall that will then cause lysis of the cell. Okay, so without this enzyme, the cell wall bursts and the bacteria dies. So it doesn't kill it actively, it weakens the cell wall. Those weak leakages in the cell wall mean that it will have a pressure gradient where water will run in and rupture the cell. These are our penicillin structures. As I said, there's been a lot of work on penicillin done semi-synthetically from the original penicillin because of bacterial resistance. So we can modify the side chains of the original beta-lactam ring to increase solubility or perhaps impart bacterial enzyme resistance, so sometimes in order to try and give resistance to beta-lactamase. So we can modify at this region here, or we can modify at this region here. What we can't touch is this beta-lactam structure because that's critical to its function. So if we alter the R group out here, you can see in penicillin G, which is the naturally occurring one, it has the R group as a CH2 out to the benzene arene ring. This is destroyed by stomach acid though, so we need to put some alterations into it to give it oral bioavailability. So if we look at penicillin V, it has this modification. It now has an ether linkage in the chain before the arene. Ampicillin, which a lot of you have possibly taken, or amoxicillin, which is a broad spectrum antibiotic, okay, they have this NH2 through to the arene. This has an amine and the hydroxyl, making it a phenol group out here. Methicillin has these methyl ethers, methyl ethers, so methoxys, on the end here. So modification of this R group, as long as we're not touching this main beta-lactam ring, will actually give us different resistance to bacterial enzymes and different resistance to our digestive system. So these modifications are generally done through semi-synthetic procedures. Okay, so ampicillin is made by replacing the side chain with other groups, and that is typically done using medicinal chemistry. Okay, so this is all to reduce the occurrence of penicillin-resistant bacteria, so making them resistant to the enzymes that penicillin-resistant bacteria produce to break down the beta-lactam ring, or to reduce the impact of stomach acid on the penicillin molecules so that it will reach its target still intact. Sometimes penicillins are also administered in combination with things like clavulanic acid, okay, because this will help to make them resistant to enzyme degradation. This clavulanic acid can bind into the enzymes, basically blocking them and reducing their activity, protecting the penicillins from breakdown. Okay, so that's a lot of information about penicillin. Key takeaways are that penicillins all work by the beta-lactam ring. You need to be able to give a description and rundown of how the penicillin works. So the primed beta-lactam ring binds to the bacterial cell wall, preventing cell wall crosslinks from being made as the bacteria reproduces. As the cell wall cannot be made properly, this causes a breakdown in the cell wall and the bacteria dies due to osmotic pressure. So you should be able to give a description of that. Most of the questions that we'll see on the exam will ask for things like that, or to identify the differences in functional groups due to the side chain and why we might use them. So make sure that you've got that in your notes, and we'll look at some questions when we go through this in class.